one of the most fun things I've done in the last 20 plus years is what I'm going to talk about today. So my talk is basically about using X-rays, synchrotron X-rays to study insect physiology. And the outline of my talk is as follows. And first, I'm going to talk about why we study bugs, some conventional techniques, explain the differences between conventional sources and synchrotrons, and then go into 3 examples of discoveries that we have done over the past years. So the first thing is, why study insects, right? Why study bugs? Well, bugs make up more than 50% of all the species on Earth. And it should be pretty obvious to everyone that they play a critical role in our ecosystem, right? And so here is a pie chart of all the species, and us humans are in this little chordate, so in this little sliver, okay? There's also a lot of bad bugs around, okay? Malaria kills more than 500,000 people a year, okay? Just malaria alone. There's this other bug called a triatomine beetle, and it kills more than 10,000 people a year. Bugs also do some very bad damage to agriculture, for example, or to plants. For example, well-known emerald ash borer. USDA estimates that so far it's done more than $10 billion in damage. Another example is this glassy wing sharpshooter, which is responsible for something called Pierce's disease that affects grapevines. And every year, the wine industry loses about $10 to $20 million to this bug. Of course, insects also do some great things. And it has been estimated that the pollination aspects of insects is worth some $200 billion a year, so which is about 10% of the total agricultural value. They also eat bed bugs. Another reason to study insects is that there is a field called biomimetics. And this is where we want to learn from nature. Okay? And there's many examples of this, not just in insects, but across all of nature. So for example, the lotus leaf is being studied for something called superhydrophobosity. The color from the butterfly does not come from pigments. It actually comes from the fine nanostructure of the wings. Okay? There's a group in Japan who is developing painless needles based on how a mosquito bites you. Because when a mosquito bites you, you normally don't feel it, because otherwise you get smacked. All right? The gecko adhesion is pretty well known, and also the Velcro from birds. Now, because there are so many insect species, that means that they have evolved many, many different solutions to life's challenges, to the physiology of life. Okay, so they've adapted to many different ways of doing things. And so it's interesting to study them and see if we can learn something from that. So what are the conventional techniques to study insects? Well, the most common is, of course, cut it open, right? And unfortunately, usually this is not so good to try to study physiology because the animal isn't kicking and alive and is somewhat dead. Another example is you can look from outside, see what it's doing. Like in this particular example here, which is actually what I'm going to talk about later, you can look at how this animal sprays from outside. Okay, you can look at it to the spray from outside. Another example is you can monitor input-output. So here, where you can look at the gas coming out, the gaseous exchange, you can put in air with no CO2 and track CO2 coming out, for example. That tells you a little bit about the respiratory system of the insect. And there's another example where here, believe it or not, biologists are detecting the electrical signals from muscles inside the animal. Okay? So this is called electromyography. And so these are some of the ways that people can use to study insect physiology. And from these, you infer the physiology. You infer how things actually work. Because until recently, you cannot really see. You cannot see how the inside is. Okay? Unlike us, you can go and you can see your lungs moving. You can see your heart in the hospital. There's no such equivalent technology. All right. So what do you need to visualize the physiology inside a sort of a small millimeter to centimeter size opaque animal? Well, you need a probe to see inside the opaque animal, right? You need good spatial resolution. And what you need is, well, spatial resolution in the 1 to 10 microns. 1 micron is 1/1,000,000th of a meter. And as reference, your hair is about 100 microns in diameter. You also need good time resolution. What that means is, physiology involves a lot of moving parts, right? So if you want to take a picture of a moving part, as you know from photography, you need a fast exposure. So that means that you need to be able to do time resolution. And then you need sensitivity to soft tissue, right? You want to see what you can, to be able to see things that you need to see. So what are some of the common biomedical imaging techniques? There's ultrasound, there's magnetic resonance imaging, MRI, and there's X-rays. So these are very typical of what you can see in a hospital, right? So how about ultrasound? Now, ultrasound is fantastic, and almost everybody in this room has probably seen this fantastic pictures, exciting pictures, right? Now, can we use this to look at little insects? Unfortunately not, because of poor resolution. Okay, the resolution of ultrasound is about 100 microns, which is not good enough to look inside the organs of the animal. How about MRI? A typical hospital MRI gives fantastic imagery of the human head, for example. Can you use this? Well, not quite. Again, the spatial resolution is pretty poor. And more importantly, it takes a long time. You have to stay still for 30 minutes in this case, okay? Now, there's people who have developed specialized MRI, and I love this picture here. It looks exactly like the previous one. Oops. Like that, except that this is made for a little pupae, which is about 2 inches, all right? And with this, you can see inside the head of the pupae, okay? You can see the brains and the lobes. Well, the problem with this is the spatial resolution is not that great, 50 microns, but the problem is it takes 3 hours. And the reason this can be done is because this is a pupae. The pupae is sort of dead, right? It's just sitting there, okay? And so this would not work for a live moving animal. Then there's X-rays, discovered some 120 years ago by Rankin. And this is a very famous picture where, his poor wife's hand was on there, and that's the wedding ring. Of course, today you can do much better in a hospital. And almost all of us have taken, X-rays of some sort of the other. Now, why can we not use this? The problem is that in a hospital, your spatial resolution is not very good. It's 100 to 1,000 microns. And there's very little contrast to soft tissue. You can only see the bones. You really can't see anything else. All right, so now I'm going to explain a little bit about the differences between X-ray imaging using conventional sources and at a synchrotron. So I think everybody here probably knows synchrotrons are facilities where we produce very high intensity X-ray beams. In fact, the first dedicated hard X-ray synchrotron in the US is the light source here at BNL, NSLS. And the next one was at Advanced Photon Sources Argonne, where I worked up until recently. And of course, the latest one is what we have here, the NSLS-II. Now, let me take a brief detour to talk about my actual job here at NSLS-II is actually to build the FXI beamline. And this beamline, the imaging capability of this imaging beamline is not the same as the imaging technique I'm going to talk about today, but it complements it, okay? So this imaging technique on the FXI is for looking at very, very small things, about 40 microns big. But the spatial resolution is very, very small, 0.03 microns, okay? And so this has incredibly good spatial resolution, but it's not so good for looking at living things, all right? And it's very good at, for example, inoperant energy storage devices and things of that nature. All right, so let me flip back to X-ray imaging again, okay? There's 2 primary contrast mechanisms for X-ray imaging. The first is what we call absorption contrast. So let's say you have a synchrotron X-ray source. Here's the X-ray beams coming out. Well, what would you look like on a detector? So let's say the detector is dark, and so when the beam hits, you see this bright stripe, okay? So let's say this is where the beam hits. So if you put a sample in like this, if the sample absorbs a little bit, weakly absorbing, then where the sample is a little bit darker, just as I've shown here. And if the sample is very highly absorbing, then it's even darker, okay? So this enables you to see if there is sample or no sample. In a conventional X-ray source, the X-ray source size, as I've shown here, is very big relative to the synchrotron source size. So again, if you don't have a sample, it looks like that. If you put a sample in, is exactly the same thing happens, okay? So there's no difference between, for absorption contrast, there's no difference between conventional and synchrotron X-ray imaging, okay? Now, there is another contrast mechanism which we call phase contrast. Sometimes, well, some version of it is called refraction contrast. So again, let's say you have a synchrotron beam, all right? If you have no sample, this is what it looks like before. When you put in a sample, something happens. Right at the edge of the sample, right there, okay, this ray gets refracted, okay? It does this. In other words, what happens is that you will have a region that now has this red and blue X-rays, so it's going to get brighter than usual, and where this ray used to go is now dark because there's no X-rays there, all right? So you'll get this bright and dark lines. So when you put a sample in, as before, the weakly absorbing sample, in this region is still absorbed, but then at the interface here, you see there's this bright and dark lines, okay? Now, if you compare this with what we had before for the absorption only, you can see that it's much easier to detect that there is something in the beam with this, right? It's much easier to see. So phase contrast greatly enhances the edges of your samples, and it's very useful for looking at weakly absorbing sample. And this mechanism is really just the X-ray equivalent of what you see every day, the visible light refraction, okay? Nothing special, just with X-rays. Now, why doesn't this happen for a conventional X-ray source, you ask, right? Well, here's a conventional X-ray source, and here are all the rays that hit near the edge of the sample, okay? And does refraction occur? Yes, it does. So you're going to do this, all right? But the problem is now, you see that there's really not much change here because there's only a lot of rays come from all over the source for every point here, okay? So because the source size is so big, basically this bright and dark fringe that I was talking about gets averaged out due to the source size, okay? So when you put in a sample, you don't see those dark and bright fringes, as I said before, okay? And that is because of the source size effect. The large source size averages out all these bright and dark fringes. All right, so as an example of this phase contrast, here's a picture of an ant head. And here's an absorption, X-ray absorption image of the ant head, okay? And here is a phase contrast. So you can easily see that this is much clearer. You can see all the beautiful air sacs and the trachea system in the air, in the ant head. Now, this technique, although I'm going to talk about, is going to be on insects. The same technique has a lot of other applications that we use it for. For example, here is a case where we are looking at fatigue cracks, okay, in a nickel superalloy. Nickel superalloys are the material that are used in turbine engines, okay? And we are tracking cracks. And you can see the cracks growing here as a function of a fatigue cycle, okay, as an example. Another example is, we're tracking in the earth science or environmental science area, we are tracking liquid flow through a column of sand. So here is the column of sand before anything, okay, in 3D. And here you can see the dark areas. This is where the liquid has come in, okay? And then in the final one here, this sand column was not packed very tightly. The liquid has gone down, and it dragged some of the grains with it, creating a cavity, all right? So, but you can imagine that this has applications in environmental science and in oil discovery and so forth. And there's another application that we have also done, which is to look at fuel injectors. Okay, so this is a diesel fuel injector, and what we are looking at is internal dynamics of this fuel injector. And what we can actually see, I don't know if you can see it, is that there's a wobble in this. Okay, and this affects the efficiency of the engine and the environmental and the pollution level of the engine. Okay, so this is just some of the more other applications of the same imaging technique. All right, so let me come back to insects. So the first example I'm going to talk about is how some insects breathe. The basic motivation here is simply to understand how they breathe. You know, this is done by, led by some of my collaborators at the Chicago Museum, Natural History Museum, and this was done some 12 years ago. So the first thing I want to explain is what are the two major modes of fluid transport in living organisms. So the first mode of transport is diffusion. Diffusion is very simple. It's just something that goes from area of high concentration to low concentration, and it happens just from thermal energy of the particles, okay? And it's a slow, random motion, and you don't need any pumps or anything like that. It happens all the time. If you're sitting in a still room and somebody has perfume on the other side, eventually you will smell the perfume, okay? The other way of fluid transport is convection, and this is a fast and forced motion where we call it active because you need some pump mechanism to push this particles from one end to the other. For example, your heart. Your heart moves blood around by convection, okay? The pumping of your heart. So what is the biology question here? The biology question is simply that if you are an amoeba, you're very, very small. You're only about 10 microns or so. So it takes very, very little time for oxygen or carbon dioxide or food to go from one side of the animal to the other side. So diffusion, which doesn't need any additional energy to do, is very sufficient for all the necessary fluid transport in a living organism. So if you're very small, diffusion is sufficient. If you're a human, like us, you're a meter or two, it's not sufficient. That's why you have the diaphragm that you have to breathe. Your lungs is a pump. Your heart is a pump, okay? So if you're big like us, you need convection. If you're very small, you don't. Well, so the question is, if you're in between, do you need? What happens? So before I explain anything, I need to say a little bit about the insect respiratory system. Insects do not have lungs. They have a network of air tubes that go from outside of the body directly to the cells themselves, okay? So this network of tubes terminate on the exoskeleton where there's a valve there called a spiracle, and then the tubes go finer and finer and finer and eventually directly feeds the cells, okay? And this is the standard textbook for entomology, the insects. Is a very old book and has been in the 4th edition here. If you cut open one of these air tubes, here's the inside and here's outside, okay? Here's the inside. You'll see these things called ctenidia. Basically, these are reinforcement structures, okay? And you can see them here, okay? They're these rib-like structures. And so if you look in this textbook, this is what it says. The ctenidia cuticle is glutterized, which means that this part here is hardened, okay? So it's like reinforcement of a garden hose. Trachea is circular in cross-section and resists any change in form because of its ctenidia, okay? Ctenidia to prevent the collapse of the trachea if pressure within a tube is reduced. So the textbook view of this is that the trachea is a reinforced stiff tube, okay? So if you look at the animal with X-ray phase contrast imaging, here's a beetle. It's about 2 centimeters. And if you zoom in, okay, to the head, and you can zoom in some more, what you see is you're able to see very clearly these tracheal tubes down to 5 microns in diameter. Okay, so you can see the entire network of air tubes in this insect very, very clearly. So what is the experiment I'm going to talk about? Well, remember I said one way to study insect physiology is by tracking the input and output. So this is what we do here. We put air with no CO2 into a box, sealed box with the animal, and see how much CO2 is coming out. This tells you about the respiratory metabolic rate of the animal, right? Just like us breathing in oxygen, breathing out CO2. And then, so this is what it looks like. The animal is in this little container here, and then we'll monitor the air in and air out, and then we'll just do with X-rays at the same time. So what I'm going to show is a movie. And so here's the animal, and we're going to be looking at this area. This is what we call the thorax, and this is the head up here, okay? So this part here is the head, and this part here is sort of the chest area right here, okay? These are bubbles in the gut. The insect swallows air, and this is what you're seeing, okay? And then the rest of this tubes here, these are the tracheal tubes I was talking about, all right? So what did we see when we first did this? Okay, so you can see very, very clearly there's a global compression of these tracheal tubes. And this was a big, big surprise to everyone, okay? So now that we know that this thing is compressing, how do we know it has anything to do with respiration? Okay, so here's the next slide, and it's a little bit complicated, so I'm going to go slow on this. On this top area here, it just tells you where I'm looking on the animal. Mesothorax is the chest, middle chest, upper chest, the femur, the legs, the head, okay? This trace here, this plot trace, is CO2 output from the animal, okay? Remember, we are monitoring the gas coming out of the end of the chamber with the animal. So this is sort of CO2 from the animal, okay? This green bars here is when every time we see on the X-ray movie, the trachea compresses, we put a green bar. When the trachea reinflates, we put these gray bars that follow it, okay? So it's compression and then reinflation, compression, reinflation, okay? So what you see here is that, so again, this is a CO2 trace, okay? The green bars are compressions, okay? And what you see here is very, very clear that every compression is associated with a spike in CO2, okay? Every compression is associated with a spike in CO2, okay? So this proves that this tracheal compression has a respiratory function, okay? And this is totally new, okay? Nobody knew this. And so remember I told you about this textbook, 4th edition was this, that I quoted from earlier. And the 5th edition, published in 2013, now has this few sentences here. Thanks to synchrotron, X-ray synchrotron imaging allows visualization of tracheal system living insect. We now know that many trachea and variety of insects compress substantially, apparently producing tidal ventilation. So this is really, really gratifying because, we did some work and it actually changes textbooks. So this is great. All right, the next thing is how butterflies drink. So the motivation of this is my collaborator is in the material science department and is interested in developing some device that can extract liquid from very, very small spaces, and for example, intracellular fluid. And it's based on a publication we had in 2012. So as you can see, butterflies drink from an assortment of sources, including wine. And I think that's really cool. And a term that you're going to hear is proboscis, and proboscis basically refers to this long sucking mouth part of the animal. Okay, so it's basically this long sucking mouth for the butterfly. All right, so what do we know about butterfly today? So the world authority on this is Austrian, I suppose. His name is Harald Kren. And in the annual review of entomology, and this is a whole chapter on feeding mechanisms of these butterflies, this is what it says. I won't go through the whole thing, but in red you can see it says the suctorial proboscis encompasses a fluid-tight food tube. Fluid uptake is accomplished by action of a cranial, the head, sucking pump. All right? So in other words, it's a straw and a suction pump in the head. Okay? So the butterfly, the current understanding is a straw and a suction pump in the head. So if you cut open a butterfly, okay, and this is what you'll see. This red are muscles that is attached to like a piston, okay? And this is a chamber, and this green is compression muscles. So there is a muscle that lifts this roof up, and then there's a muscle that pushes it down, just like a syringe, nothing more than a syringe, okay? So this is what you see, and you can see that this is not too long ago. So we'll do the experiment. Here's a butterfly, and what we're doing is we're feeding it some sugar solution, okay? And then we just put some x-rays through it. All right, so let me orient you to this movie. So here's the head of the butterfly. This is what we're looking at. And the proboscis is down here, just like I've shown here. Okay, and the body is sort of up. All right, so this is the head. All right, so here's the pump. Here's the pump that he was talking about, right? And this is what we thought was exactly according to the textbooks. So from this, we're able to quantify a lot of stuff that was previously unknown. We can quantify all the fluid volume and speed and so forth. So basically, at that point, we thought that, okay, this is how it works, right? So there's a syringe, right? And there's two valves. There's a valve at the back of towards the back, and there's a valve in front. So in the beginning, this back valve is closed, and you're basically pulling the syringe stem up. You're sucking the liquid, okay? You're sucking the liquid into this chamber. And then this valve will close, okay? And then your compression muscles, so now you're pushing the syringe out, and the back valve opens, and the liquid goes to the back, all right? So it's basically a syringe with two valves, okay? And this is totally consistent with, what the current understanding was. And so Jake Soha, who was my postdoc back then, he's now a faculty at Virginia Tech, we thought, okay, the story was over. And I was at a conference just shortly after that, and I saw this. Butterfly proboscis as a microfluidic system. Microfluidic just means control of small amounts of fluids in a biomedical context. And you read this, it's very complicated. And so I talked to the speaker and her advisor after this, and I said, no, it's a straw and pump, and I showed him the movies and everything, But again, this is one example where I think why you go to conferences, right? It's for these few magical moments that you get. So anyway, after a long discussion with this group, basically, what he said was, look, butterflies can feed from damp surfaces. In fact, butterflies have to feed on dung because they actually need some amino acids that apparently can only get from dung. And they also feed from, bananas, which is not a pool or liquid. Now, why is this a problem? So let's put it in human context to see why this is a problem, okay? So a butterfly weighs about 1 gram. A human weighs about 80 kilograms. I think an average US person is 80 kilograms. So the human is 80,000 times heavier than a butterfly because mass is the length to the third power, okay, which is the same as saying, if you're a solid ball, the ball twice as wide is 8 times heavier, right? So in terms of the length of a human should be cube root of 80,000, which is about 40, all right? So let's see if that's true. Well, a butterfly is 25 to 50 millimeters, and 40 times of that is about, 1 meter to 2 meters. It's pretty close. The proboscis of a butterfly is about a centimeter, so the human equivalent will be about 40 centimeters. The diameter of the proboscis is 0.1 millimeters. The human equivalent would be about 40 millimeters. It's 4 millimeters. So this is about the size of your standard straw. Okay? So in other words, the butterfly's proboscis, in human terms, is just like a straw about 40 centimeter long. All right? So you can ask yourself, can you suck liquid from a banana using a 40 centimeter long straw? Right? So clearly there's a problem with this straw model. So here are some pictures of butterfly feeding. Now, there's something unusual about this. Okay? The unusual thing about these pictures is this. See how this tip is curled like that? It's not like how you would do a straw, right? You see this, it curls inside like this. So the dorsal, this is how it feeds. If you look at the electron microscope picture, SEM, here's the proboscis. If you do a section across it, you see that there's two halves, and here's a food canal in between. Okay? Here, this is where the food comes. And here are the two halves. And if you look down this way, you see there's actually a zipper-like mechanism. So the butterfly proboscis is actually made from two parts that are zipped together. Okay? This, among other things, helps it to be able to curl up. Okay? Because this space allows it so that the outside, right, is a little bit longer, than the inside, so it enables you to curl up. Okay? So the proboscis is not a solid tube. There's two parts held together with these zipper-like structures, as you can see here. Okay? And there. Okay? So those are the zipper-like structures. And you can actually measure that there is some space in between the zippers. And you can sort of see it here, right? And to be expected, the zipper is not liquid tight. And so, again, here is a cross-section of the zipper, of the proboscis at 260 microns from the tip. Here's food canal. But as you go closer to the tip, what you see is that this food canal closes. In other words, this food canal narrows, and at the tip is actually no food canal there. It's actually closed. So the butterfly proboscis is clearly not a simple straw. So here's an experiment that we did. And this is the kind of experiment like before that can only be done at a light source in a synchrotron. We took a butterfly, and we carefully unfold its proboscis and put a drop of liquid there and image it. So here is the proboscis. The head is there, just like I've shown in this orientation. And here's the food canal, and just put a drop of liquid there, okay? And what do we see? So what you're seeing is very clear that it can take the liquid not from the tip. Clearly it's not from the tip, right? It's just from the top surface of the proboscis. Okay? Okay, so there's two key physics concepts I want to talk about here. One is capillary action, and you see this every day, paper towel sucks up liquid. And it's just that this capillary action just describes the fact that narrow pores or channels suck up liquid. And the smaller the channels, the stronger the pull. The other is something called the plateau instability. And the plateau instability is a tendency for liquid streams to break apart. Again, you can see this with a tap every day. So these two physics principles are actually what the butterfly uses. And I'll show this. So from those movies, we can measure the dimensions, for example, of the food canal. And from those these liquid bulges, we can measure L, the lateral extent of this liquid bulge. The plateau instability from the theory tells us that When L is about 2.8 pi r, that's when the instability will occurs, okay? Now, we can measure L, left side. We can also measure R independently. So we can see if the left side and the right side agree. And our measurements show that the left side here is 188 plus minus 9, and the right side is 15 plus minus 34. And so within the errors, you can see that they agree pretty well. So the measurements is very consistent with this plateau instability theory. In other words, what we think is happening is the following. Here is the food cavity. You put a drop of liquid here. These are the very narrow spaces between the zippers I was talking about, okay? So what happens is that the liquid is drawn in by capillary action to coat the inside surface of this food canal. The plateau instability initiates these bulges. These bulges grow and merge into what we call liquid bridges, and then the head pump sucks those in, okay? All right, so that's how the butterfly drink. And finally, an example, this is the latest example of work that we've done very recently. It's about a bombardier beetle. This work was covered in quite a few mass media, and the headline I like best is from Washington Post. It says, this beetle's butt is basically a machine gun. So the motivation for this is how to design better armor against explosions, and you'll see why shortly. Why is the bombardier beetle, and yes, this is the publication in May this year in Science. So why is the bombardier beetle very interesting? This beetle can shoot a spray of about 100 degrees Celsius of noxious liquid at its prey, okay? And this liquid is actually pulsed. Its pulse is something like 500 pulses per second, okay? So that's why Washington Post says it's a machine gun, all right? So this beetle, when you come up, and actually what you see here is this guy is trying to have it spray, so he takes a pair of forceps and grabs his leg and he zaps it, okay? All right, so how does the beetle do this? If you cut the beetle open, this is what it tells you. There are glands that produce a chemical that is stored in a reservoir. There is a reaction chamber, and between the two, there is an entrance valve. And then, so the idea is that the chemicals in here, when come in contact with enzymes that reside in the reaction chamber, the explosion happens, okay? And so if you cut the animal open and you extract them, this is what it looks like. Here's the reaction chamber. Here's the reservoir, okay? Those are the reaction chamber, and this is the rear of the animal, okay? So the noxious jet pulses come out here. All right, so you can see that they're not very big. It's less than a millimeter across. And one thing I want you to notice, this sort of funny Mickey Mouse-like ear-like structure of the reaction chamber. So prior to us doing the work, what do people know? Well, again, this is done by external observation. They can measure the spray temperatures. Like I said, it's up to about 100 degrees C. They can measure the speed. They can measure how far it goes. And they can also, by doing quite smart, they use audio recording in the early days to know that it's actually 500 pulses per second, okay? The question is, how do they make these pulses? And there are actually three theories. One, going back to this picture, one is that this entrance valve here opens and closes at 500 times a second, right? Very fast. So that's first theory. The second theory is that this entrance valve has a mechanism where at every explosion in the reaction chamber, it forces itself closed. Okay, so it has a self-closing mechanism. And the third theory is that when the liquid chemical reactants in here enter this chamber, there's a chemical reaction happens, but there's no explosion because what happens is that it creates a superheated fluid state, all right? So this is just like, sometimes you take your cup, you microwave the water, you take it out and you see nothing, it looks very flat, there's no boiling, but then when you dump your tea bag in it, you go, like that, right? So this is the third theory, which is the explosions happen by control of this exit valve, all right? So those are the three theories about how the pulses are produced. So, okay. All right, so here's the visible light picture. Here's the insect. It's held by putty. And what we're going to do is try to get it to zap and could you see that? That was a big sort of a flash of stuff. It's hard to see. Okay? So this is what it looks like from the outside. Okay, what does it look like on the inside? So here's the beetle. I'm going to be imaging X-ray wise near the butt here. Okay, here's the butt. All right, and here are all the air tubes. At first, you cannot see anything. So these images are acquired at 2,000 frames per second. This can only be done at a synchrotron. Okay? What I'm going to play back to you is 5 frames per second. So what you see now is 400 times slower. Okay? And there's those reactions. Those are those explosions. Boom, boom, boom. Okay? And you can see this Mickey Mouse-like structure I was telling you about. So the vapor goes into the entire reaction chamber. So based on this, we knew we could see that the vapor occurs inside the reaction chamber, and we know that this superheated fluid hypothesis was incorrect. Now, the final video I'm going to show you is actually the most impressive, in my opinion, the most fantastic insect video we have taken in the past 12 years at Argonne, all right? This is again done at 2,000 frames per second. Playback again is 5 frames per second. Same thing. Now, in this clip, the animal has already fired before, so there's a vapor pocket in the reaction chamber before we started, okay? So here you can see, here's a vapor pocket. Now, it's going to be very hard to see, so I want you to focus sort of here, okay? Could you see that? So what you should be seeing is a drop of reactant entering the chamber. Okay, watch there. I don't know if you could see it. Well, if I slow it down and go frame by frame, this is what you will see. Okay, here's where it started. I think in these couple of frames is very clear. Right, here's the liquid drop coming into the reaction chamber. So this is fantastic because with this, we can quantify the chemical reaction. We know exactly how much chemicals are involved in here, okay? And so this is really, really a stroke of luck because we would not have seen this if this animal didn't have this vapor pocket before, okay? So let's put it in context. So we have done the calculations and each explosion releases about 4 millijoule, and it's equivalent to 1/1,000,000th of a gram of TNT, not very much. Acceleration rates in there are tremendous. 10,000 G is the acceleration that we measured inside. Okay, as in context, the maximum human on a rocket sled is about 45 G. So this is, people cannot survive this. So it's definitely a huge explosion that occurs inside this insect. To put it in human context, a beetle weighs about 50 to 100 milligrams. Humans, again, 50 to 100 kilograms. So we are about 1,000,000 times heavier than this beetle. So the amount of equivalent TNT in this explosion is a millionth of a gram of TNT for the insect. So for us, it'll be about a gram of TNT. That doesn't sound like a whole lot, except that, gunpowder in a bullet is only about 0.2 to 0.3 grams. And actually, I found this cool video. So this guy drilled a hole and put in equivalent of about a gram of TNT in this wooden block. And this, I have to acknowledge this video, and see what happens. Right. So this is just 1 gram of TNT. So that's why my collaborator at MIT is very interested in this, right? Because this insect has this little chamber in its belly, not belly, but in its body that can withstand something like this. It's phenomenal. All right, so in conclusion, synchrotron X-ray imaging have really opened the doors for studying insect physiology. This was just not possible before. And as I've shown you, the most gratifying thing is that our results have really upended long-held textbook views. The technique is very broadly applicable, and I showed you a few examples of it. Real-time visualization of materials, fabrication, fatigue, and failure, in operando studies of energy storage devices, in vivo tracking of fluid through soils and cryo rocks. NSLS-II's beam properties are ideal for this kind of imaging. And the small source size means that we get sharper images, a higher phase contrast. The high intensity enables to do faster and faster imaging. And as an example, this is an image taken at HXN beamline, which is not optimized for this kind of work, but nonetheless, the power of NSLS-II can be easily seen here. Here's the butterfly proboscis, and you can see, I don't know if you can see it very clearly, you can see all kinds of little hairs. It's a beautiful image here, okay? So the ability to do this in NSLS-II would be fantastic. So finally, let me acknowledge, as I said, this insect work was done at Argonne National Labs before I came here, and my collaborators are Kamel Fizar and Jake Soha, and Mark Wesney for the insect breathing work. The butterfly work was done with Konstantin Kornev at Clemson, and the bombardier beetle was done with Christine Ortiz at MIT. And with that, I thank you, and I'll take any questions.
We have time for a few questions. Anybody would like to ask Wah-Keat something?
Yes.
That's so neat. Why would you want to study batteries?
I'll have to take that offline. No, we actually do a whole lot. But this is a lot more fun to talk about than batteries, to be honest.
The answer is it's all about the money.
Thank you. Yes?
Radiation damage is a big issue?
Very, very good question. And that's almost, so radiation damage is an issue. And so prior to a lot of this, we actually did a lot of studies on what can we see and how long can it survive. But as you can see in the examples I showed here, that physiologically they can function, right? Like drinking, the butterfly is drinking, right? Breathing, you can see the insect is breathing. And you can, but it's true, you cannot, what we have a lot of information on how long we can zap it on the head, how long we can zap it in the abdomen, and so forth. Yes, but that is it.
So after the experiment, they all die?
No. It depends on how long you zap it for. Yeah. So if you take a beetle and you only zap its abdomen, you can zap it for hours on end. When you take it out, it'll just run away. But if you zap it on the head for a long time, then it doesn't do so well.
So on the first movie of the bombardier beetle, you don't see the reaction chamber in the image until the fluid has moved into it. Is that because the fluid is important for the refractive index of that?
Yeah, so in order to see this refractive index, you still need a electron density change, right? And so what we are seeing when the chamber is filled with liquid, the density differences between the chamber and the liquid is too small for us to see. So we only see it when the vapor happens, when the reaction and creates the vapor. Yes?
The optics you use, is this just straight projection from a point onto a detector a long ways away, or do you have a lens there?
No, this is just pure propagation.
Yeah.
Yep.
Does anybody know what the chemical signal?
Yes, yes. So that's actually very well known, and that's why we can calculate the energy and all the heat related quantities, yes. They are hydroquinone. Yes. And then there's a bunch of enzymes in the reaction chamber. And so when the hydroquinones go into the reaction chamber, that's when the chemical reaction happens. Yes.
What limits the frame rate for your acquisitions?
So these images were done at 2,000 frames per second, and we were using monochromatic light at the APS. And so what limits it is how much the X-ray intensity is. So if you want to go faster, then you need more X-ray intensity. And so, for example, at let's say 10 keV, NSLS-II will have 10, 20 times more photon density. And then that would mean that you can do it at much higher frame rates.
Detector isn't limiting at all.
So the detectors can actually go faster because so these days you can buy these high-speed cameras with several thousand frames per second. And if we only don't use the entire 2K by 2K, you can go fairly high. Yeah.
If there are no further questions, we invite you all to join us for refreshments out in the lobby.